Hello everybody, this is Jyoti and this is the lecture 1. This lecture series is all about investigating and manipulating materials in the nanoscale. So basically in this lecture series I am going to talk about what are the different characterization tools with which we try to find out the properties of nanomaterials. So first thing which comes in our mind is what is observation. So observation, basically it is the key to making new discoveries. If you have to discover something new, basically you have to observe. And this is specifically true in the nanoscale. And these investigations are not possible without observations. How the observations can be done? So at the nanoscale, these observations basically are done with a probe, and this probe, it may consist of either photons, electrons, neutrons, atoms, ions. At times it may be atomically sharpened. What is the frequency of probing light or a particle? So the probing particle or light has varying frequencies ranging from gamma to infrared rays or beyond in case of photons, or hyperthermal to relativistic energies. How the resulting information is processed? So whatever the resulting information which we get after probing that material with the help of this electrons, photons and all, that information, it may be in the form of images or it may be in the form of spectra. And what kind of information we get from these spectra or images? So here the information, it may be topographic, it may be geometric, it may be structural, it may be chemical, or it may give you physical details of your sample. What we use to see objects in more detail? So basically what we use is microscopy. Now certain points we have to note. The best distance that one can resolve with optical instruments disregarding all aberrations is about 0.5 lambda, or it is of the order of 250 nanometers with visible radiation. All forms of microscopy are aimed at improving our capacity to see, and under ideal conditions, the smallest object that the eye can resolve, it is about 0.07 nanometer. Now this limit is related to the size of the receptors in the retina of the eye. So types of microscopy, basically we have optical microscopy, electron microscopy, scanning probe microscopy and others also. So what is a microscope? So microscope, it is an instrument which is used to form enlarged images, and the microscope, it consists of two Greek words, micros, that means small, and scopos, that means to look at. So Antony van Leeuwenhoek discovered the first microscope, and this single lens microscope had to be held against the eyeball because of their short focal length, and they helped in the discovery of bacteria. In fact, he was the one who invented more than 500 different types of microscope, but then he didn't get the appreciation he thought he will get, and that is how he destroyed most of his microscopes. Yeah. Now this is very important term. Whenever we are dealing with microscopy, basically you should know this term, resolving power. So one is resolution, second is resolving power. So the resolution achieved by a particular instrument under optimum conditions, while resolving power, it is a property of instrument and it is a quantity that may be estimated. Resolution is equal to or poorer than the resolving power and has to be determined by the instrument. So resolution is basically how separate images you can see of two closely spaced objects, but resolving property, it is a property of instrument and it can be estimated. Okay. Now kinds of microscopies. So when we talk about microscopies, basically we talk about two. One is transmitting, second is scanning. So transmitting, in this the probe beam passed through the specimen is differentially refracted and absorbed, and while in case of scanning, the probe beam is scanned over the surface and the image is created point to point. So I guess this important terms was the earlier side. It came here, that is okay. Yeah. So kinds of scanning microscopes. Scanning electron microscope. So a monochromatic electron beam is passed over the surface of the specimen, which induces various changes in the sample, and the resulting particles from the sample are used to create an image of the specimen. Now this information is derived from the surface of the sample. Second we have is scanning ion microscope, SIM. So in case of SIM, what we use is charged ions to obtain the image, and the process it etches away the top surface. Third we have is scanning acoustical microscope, SAM. This uses ultrasonic waves to form images. The best resolution achieved is of the order of 2.5 microns, which is limited by the wavelength of sound, and its advantage is that it allows one to look at biological samples. Scanning light microscope, SLM. A fine beam of visible light is passed over the surface to build up the image point by point, and it facilitates increased depth of field and color enhancement. And last one is scanning confocal microscope, SEM, and a finely focused beam of white or monochromatic light is used in this case, and more commonly this technique is called as confocal microscopy. Thank you so much. Please like and subscribe.